Good afternoon, dear friends. I'm Sol from Biological Sciences with Sol channel. Today I'm with a video, and this video is all about enzymes. So please don't forget to like and share this video and subscribe my channel. So let's proceed. So this is all about enzymes. So let us first see general properties of enzymes. Enzymes are proteins that catalyze chemical reactions in biological system. As you know, there are two types of catalysts, biological as well as non-biological catalysts. So enzymes and some RNA molecules are biological catalysts. So enzymes are proteins that catalyze chemical reaction specifically in biological system. They increase rate of chemical reactions taking place within the living cell without changing themselves. These enzymes increase the speed of chemical reaction that convert the product, the reactant or the substrate to the product. However, at the end of chemical reaction, enzymes are saved or they are not changed. That means this is substrate plus enzyme, finally product plus enzyme. So the speed of rate of chemical reaction or the increase rate of chemical reaction. However, they are not changed in the chemical reaction. The other point is nature of enzymes. Almost all enzymes are protein in nature, but depending on the presence or absence of non-protein components with the enzyme can exist as simple enzyme or holoenzyme. This classification is based on the presence or absence of non-protein components in the enzyme. Simple enzyme is made up of only protein molecules not bound to any non-protein compounds. For example, pancreatic ribonuclease, an enzyme which catalyzes the breaking down of nucleic acid, especially RNA, is a type of simple enzyme. Whereas holoenzyme is made up of protein groups as well as non-protein components. The protein part or the protein component of holoenzyme is known as apoenzyme, whereas the non-protein component of this holoenzyme is known as cofactor. If the cofactor is organic compound, it is known as coenzyme, whereas if the cofactor is inorganic compound or an organic group, it is known as activator. So mostly these activators are metal ions. If the cofactor is bound tightly to the apoenzyme, that means protein part of an enzyme, and it's difficult to remove without damaging the enzyme, it is sometimes known as a prostate group. Here, enzyme can be classified into simple enzyme if it is made up of only protein, and it is holoenzyme if it is made up of protein part and the non-protein part. So this the non-protein part of an enzyme is known as cofactor, and the protein part of holoenzyme is known as apoenzyme. So if the cofactor is organic compound, it is coenzyme, whereas if the cofactor is inorganic molecule, it is activator. So to say something about coenzymes, they are derivatives of vitamins without which the enzyme can't exhibit any chemical reaction. A holoenzyme that has coenzymes as a cofactor can't exhibit any reaction without these coenzymes. One molecule of coenzyme is able to convert large number of substrate molecules with the help of its enzyme. Coenzymes accept a particular group removed from a substrate, or they may donate a particular group to the substrate. So these coenzymes are called co-substrate because the change that takes place in the substrate are complementary to the change to the coenzyme. Here in this figure, you can see that lactate or lactic acid is converted to pyruvate by the reaction, the catalytic action of lactate dehydrogenase. This NAD+ is act as coenzyme, which receives hydrogen atom. So it is converted to NAD+. Therefore, it receives a group which is hydrogen atom, and the lactate or lactic acid is converted to pyruvate. In this figure, glucose is converted to glucose 6-phosphate by the catalytic actions of hexokinase. But ATP acts as a cofactor, coenzyme, which donates phosphate group to glucose to convert glucose to glucose 6-phosphate. Therefore, coenzymes can act as acceptor of particular groups like that of NAD+, or they may donate particular group to a substrate like that of ATP. NAD, FAD, coenzyme A, ATP are good examples of coenzymes. What about metal ions in enzymes? Many enzymes require metal ions like calcium ion, potassium ion, magnesium ion, ferrous, copper ion, zinc, manganese, cobalt, and the like for their activity. So these enzymes that has metal ions can be metal activated enzymes or metalloenzymes. There are two types of enzymes. The first is metal activated enzyme, whereas the other one is metalloenzymes. What's the difference? In metal activated enzymes, they form only loose and easily dissociable complex with a metal ion and can easily release the metal without denaturation. So easily the enzyme and the metal ion can be separated without any damaging of the enzyme in metal activated enzymes. But in metalloenzymes, they hold the metal ion tightly on the molecule and do not release it even during excessive purification. So the enzyme and the metal ion are tightly bonded to each other. So such metal ions are known as prostate group. Metal ions in metalloenzymes are not releasing the protein part of an enzyme, and such metal ions are known as prostate group. How these metal ions promote enzyme action? For example, coenzymes can receive or donate particular groups from the substrate or to the substrate. But how these metal ions promote enzyme action? For example, they can maintain or produce active site or active structure conformation to the enzyme. That means they are used to make the active site of an enzyme that is complementary to the shape of the substrate. For example, glutamine synthase is such enzyme. The other is promoting the formation of enzyme substrate complex, because first the substrate to be converted into product, it must be joined or mix a complex with enzyme. So enzyme substrate complex must be formed to change the substrate to the product. Therefore, these metal ions can form that help or can help enzyme substrate, the formation of enzyme substrate complex. For example, enolase and the carboxyl peptidase are such enzymes. They act as electron donors or electron acceptors. For example, iron sulfur protein and cytochromes in electron transport chain are good examples, because in electron transport chain these molecules receive electrons and donate electrons. Therefore, metal ions in enzymes, they maintain or produce active structure. They promote the formation of enzyme substrate complex, or they can form, or they can act as electron donor or electron acceptor. And these are some actions, mechanism of actions of metal ions in enzymes. What are properties of enzymes? So first, let us see active site. Enzyme molecules contain a special pocket or cleaved called the active site. So if this is an enzyme has special pocket or cleaved where the substrate are tight. This site is known as the active site. The active site contains amino acid chain that creates three-dimensional surface complementary to the substrate. That means the shape of the active site and the shape of the substrate are complementary because the substrate are tied to the enzyme to the active site. The substrate binds to an enzyme at active site, forming this enzyme substrate complex. A substrate means a substance or a reactant on which an enzyme acts upon, makes or binds to an enzyme at its active site to form enzyme substrate complex. Then enzyme substrate complex is converted into enzyme product complex. Later on, this enzyme product complex is converted or dissociated into enzyme and product. For example, this is an enzyme and this is a substrate. They are complementary in shape. So especially at the active site of an enzyme and the shape of this substrate are complementary. Then enzyme substrate complex is formed. Later on, enzyme product complex is formed. Finally, products and enzyme are separated or dissociated. So at the end of reaction, enzymes are not consumed or they are not used up. The active site of an enzyme may contain free hydroxyl group of serine, threonine, and tyrosine amino acids, and it may contain sulfhydryl group for cysteine, and it may contain imidazole or imidazole group of histidine to interact with the substrate. When we say substrate and enzyme interact with each other, especially at active site of an enzyme, so this active site of an enzyme contains these free functional groups. That means hydroxyl group for serine, threonine, and tyrosine. They have this OH group on their side chain. Cysteine has sulfhydryl group on its side chain. Histidine has imidazole group on its side chain. So by using these side chains, freely substrated enzymes interact with each other. The second property of enzymes is catalytic efficiency or enzyme turnover number. Enzyme turnover number refers to the amount of substrate converted to product per unit time. How much substrate is converted to a product in a given time? This is enzyme turnover number or catalytic efficiency. Most enzyme catalyzed reactions are highly efficient, proceeding from 1000 to 100 million times faster than uncatalyzed reaction. When we compare the rate of reactions, that means enzyme catalyzed reaction and uncatalyzed reactions, enzyme catalyzed reactions are, especially most enzyme catalyzed reactions are more faster. They are faster than uncatalyzed reaction. Typically, each enzyme molecule is capable of transforming 100 to 1000 substrate molecules into product in each second. Within a second, 100 to 1000 substrate molecules can be converted to a product by the catalytic actions of each enzyme, single enzyme. Carbonic anhydrase is the fastest enzyme which catalyzes the conversion of carbon dioxide and the water into bicarbonate and the proton, that means hydrogen ion. So this is the fastest enzyme. The other property of enzymes is specificity. Enzymes are specific for their substrate. The specificity of enzymes are divided into absolute specificity as well as steroid specificity. In absolute specificity, one enzyme catalyzes or acts on only one substrate. One enzyme, one substrate. For example, urease, an enzyme which catalyzes the hydrolysis of urea but not urea. In steroid specificity, enzymes are specific to only one of the isomers, even if the compounds are one type of molecule. As you know, isomers are molecules which have the same chemical formula, but they may have structural difference. Therefore, in this steroid specificity, enzymes act upon one type of the isomers. For example, glucose oxidase catalyzes oxidation of beta D-glucose but not alpha D-glucose. This beta glucose and alpha glucose are isomers. They have the same chemical formula, but they have somehow structural difference. Therefore, glucose oxidase acts on beta glucose, not on alpha glucose. Arginase catalyzes the hydrolysis of L-arginine, but not D-arginine. L-arginine and D-arginine are isomer molecules, so this arginase catalyzes the hydrolysis of L-arginine, but not D-arginine. The other is maltase, which catalyzes the hydrolysis of alpha glycosidic bond, but not beta glycosidic bond. Therefore, in general, this absolute specificity and the steroid specificity are the two types of specificity of enzymes. Bond specificity is another criteria of or properties of enzymes. In previous slide, we see that enzymes are specific for a specific substrate. That means an enzyme acts upon a specific substrate, either in the absolute specificity or in steroid specificity. The enzyme acts upon a specific substrate. But here, in bond specificity, an enzyme acts upon specific bond or leakage. Enzymes that are specific for bonds or leakage, such as ester bond, peptide bond, or glycosidic bond, belongs to this group. For example, esterase. These are a group of enzymes that act upon ester bonds. Peptidase are a group of enzymes that act upon peptide bonds. Glycosidase are enzymes that act upon glycosidic bonds. Therefore, enzymes are specific for a specific substrate as well as they are specific for a specific bond. Regulation of enzyme activity is another property of enzymes. Enzyme activity can be regulated. That means enzyme can be activated or inhibited so that the rate of product formation responses to the need for the enzyme. That means enzymes are not always active, but based on the requirement of product formation, enzymes can be activated or inhibited. Whenever the product is needed, enzyme will be activated, and whenever product formation is not further needed, so enzymes can be inhibited. Therefore, they are regulated based on the requirement of product formation. And the other property is Zymogens. That means inactive form of enzymes. Some enzymes are produced in nature in an active form, which can be activated when they are required. During production, they are formed in active form, whereas later on they will be activated. Such type of enzymes are known as Zymogens. That means inactive form of enzymes are known as zymogen, or they are known as proenzymes. Many of digestive enzymes and enzymes concerned with blood coagulation are in this group. They are produced in zymogen form, which is inactive form, but later on they will be converted to their active form. Some examples of zymogens are pepsinogen. This zymogen is from gastric juice, and when it is required, this pepsinogen is converted to its active form known as pepsin. Trypsinogen is another example of zymogen. This zymogen is found in the pancreatic juice, and when it is required or it is needed, it is converted to its active form, which is known as trypsin. By the way, why these zymogens are formed? Why enzymes are formed in the zymogen form or inactive form? Zymogen form of enzyme a protective mechanism to prevent autodigestion of the tissue producing these digestive enzymes, and to prevent the intravascular coagulation of blood. If this type of enzymes are produced in active form, immediately they digest or autodigest the producing tissue itself or the producing cell. Therefore, they are formed in zymogen form, and the producing tissue as well as the intravascular coagulation of blood will be prevented. Isoenzymes or isoenzyme is another property of enzymes. These are enzymes having similar catalytic activity, act upon the same substrate, and produce the same product. These isoenzymes are enzyme types which catalyze the same chemical reaction and act upon the same substrate and produce the same products, but originate at different sites of the body and exhibit different physical and chemical characteristics such as electrophoric mobility, amino acid composition, and immunological behavior. Lactate dehydrogenase is a good example of isoenzyme, and it can be abbreviated as LDH, or simply it can be LD. And this LDH or lactate dehydrogenase is an enzyme which catalyzes the conversion of lactate or lactic acid to pyruvate. And as an isoenzyme, this lactate dehydrogenase has 5 different enzymes. These 5 different enzymes have the same catalytic activity, and they act upon the same substrate, which is lactate or lactic acid, and they produce the same product, which is pyruvate. Based on their source, their subunits, each of these 5 enzymes have 4 subunits. So, for example, lactate dehydrogenase 1 has 4H subunits, H stands for heart actually, M stands for muscle. So the first lactate dehydrogenase has 4H subunits, and this enzyme is originated from heart. The second lactate dehydrogenase is or has 3H and 1M subunits, and it is originated from red blood cells. And then the third one has 2H and 2M subunits, and it is originated from brain. The fourth one has 1H and 4M, so it is originated from liver. And the fifth one has 4M subunit, and it is originated from muscle, especially from skeletal muscle. So this is lactate dehydrogenase, which is a good example of isoenzyme. The other good example of isoenzyme is keratin phosphokinase, or simply keratin kinase. This keratin kinase or keratin phosphokinase exists in 3 different forms, each having 2 polypeptide chain, and subunits are B stands for brain and M stands for muscle. Actually, this keratin kinase or keratin phosphokinase is used to convert this keratin to keratin phosphate by taking one phosphate molecule from this ATP. So as isoenzyme, this keratin kinase has 3 different forms: keratin kinase 1, keratin kinase 2, and keratin kinase 3. This keratin kinase 1 has a polypeptide chain of BB, because it has 2 polypeptide chain, and these polypeptide chains are BB, and that is originated from brain. And keratin kinase 2 has MB as a polypeptide chain, and that is originated from heart. Whereas keratin kinase 3 has MM polypeptide chain, that means originated from muscle, especially skeletal muscle. Thank you once again for watching, and I will come back soon with another new video of the second part of these enzymes. Bye bye.